passenger pigeon was around for 6 million years. And people remembered how amazing it was that these animals darkened the sky for hours at a time.
There's these stories about these giant flocks blocking out the sun, and their wings sounding louder than thunder.
We developed the techniques to hunt and net these birds and basically kill them all. And then it was really gone. When I was growing up, my mother said when the last one died in 1914, it broke America's heart. It was one of the prideful things that America had to show the world. It was gone.
This book, this is essentially the Bible of passenger pigeon. I am currently the lead researcher for the de-extinction project to bring the extinct passenger pigeon back to life. When I say, oh, we'll bring back a passenger pigeon, everyone goes, oh my God, that's like Jurassic Park. And I think, no, it's not. Not at all.
Our scientists have done things which nobody's ever done before.
Yeah, but your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
I don't think a pigeon is going to, pluck lawyers off of toilets.
No.
So how we're actually going to go about de-extinction? Every organism has a unique code in their genes. This one would be good, I think. So these are actual DNA codes from a chunk of a passenger pigeon foot. Now, if you can get DNA, then we can actually make it into a living cell. We're going to sequence its whole genome. We're gonna build from scratch the code that is a passenger pigeon, one gene at a time, compare it to its closest relative. Then we're going to introduce DNA into the living cell of a band-tailed pigeon. And when you introduce an extinct animal's egg cell into a new mother, then you've changed the game, which has been done. The extinct Iranian ibex was successfully cloned.
That was the first de-extinction. In this particular case, the animal only lived about 10 minutes, but it was proof of concept, and with further efforts, we hope, followed by everything from the passenger pigeon to the Tasmanian tiger.
It's one of the most famous human-caused extinctions in the world. Benjamin, the last Tasmanian tiger, died in 1936. It was our direct activity that caused that extinction. For me, biotech is the future of conservation because our meddling is not unnatural. It is what species do.
One of the things that we've gotten used to is the horrifying realization that extinction is forever. But what if the new truth is that de-extinction is forever?
Hello. How are you? Not muted? We're getting better. We're getting there. Thank you for the $1 donation. Here we go. Podcast, let's do it. Is that not the coolest video you've ever seen? I may be more excited for this podcast than I ever have been at Uncle Beanbag. Try hard. Okay? I'm not gonna put it in a chat though. Thank you for the $5. We're $6 in. Like I said, guys, I don't know that I've ever been this excited for a podcast. I told you I watched these TED Talks a while ago. And it's crazy that we're actually talking to Ben Novak today. Ben Novak, you guys saw him in that video. He is the lead for the team for the Passenger Pigeon Project, and he is a de-extinction biologist with Revive Restore. So Revive Restore is where your donations are going to today. The mission statement of Revive Restore is to enhance biodiversity through new techniques of genetic rescue for endangered or extinct species. Dan with the $10. Can't watch the whole podcast, I'm sorry about that, but thank you so much for the donation. I appreciate it. And a 3-month sub. Thank you. I'm getting used to this, to the bar. It looks good though, huh? Everybody clap for the Conservation Cast team. Locke did a great job with this. It's beautiful. The alerts are updated. They look so good. They look so cool. Deja Vu, thank you for the 6 months. So. And Catsley, thank you for that. And American Eagle Foundation with 8 months. And Mr. Boom and Trap Clap. So nice. Thank you guys. Thank you. Go podcast team, go. People cheer. Yes. So today we're talking to Ben Novak about passenger pigeons. Get your questions ready at Saito. If you have questions, we have the doc open. So serious questions only, please. It makes his job and my job a lot easier. And if time permits, I will ask him those questions, but we're gonna do it like we normally do, pretty casual conversation. I'll ask him about Revive Restore so you guys know exactly where your money's going to. As always, the guest knows obviously much more than I do and is the best at explaining both their research and what they're representing. So let's just get to it, right? Should we just get to it? I left this hold. Ready? I'm so excited. I'm excited too. Okay, I'll be back in a minute. Hello. Can you hear me?
Hello. Yeah, I can hear you.
All right. How are you?
I'm doing good.
Good. Here, let's see if they can see you. Okay. Guys, oh, that's not right. Hang on. That's right. Okay. Scap. Guys, let me know how the audio and video is with all this, but let me know as we progress. So, hi Ben, how are you?
I'm doing great.
Thank you so much for coming on. I'm so excited. They all know how excited I've been. I've been talking about this for like weeks. So this is awesome. We've already gotten $16 in donations for Revive and Restore. So thank you guys so much for starting us off. Can you tell us before we start what you guys do?
That's awesome. Every dime, every quarter helps. So Revive and Restore is a nonprofit, hence why your donations are so significant to us. We're a conservation nonprofit, and we're really the only nonprofit in the world working in conservation in the space of trying to innovate and advance the adoption of biotechnology for wildlife conservation. And we're really focused on what you can get from genomics, the genome, your DNA, applying DNA technologies for every bit of improvement they can add to wildlife conservation. Our actual tagline, I think, is a genetic rescue for endangered and extinct species, and it's that extinct part that we're most famous for.
Yeah, so your research is mostly passenger pigeons, right? So how did you start? Where did you start?
So I joined Revive and Restore basically right when they started in 2012. Passenger Pigeon was the flagship project. And it's the idea of bringing back the passenger pigeon to nature.
For those watching that don't know what a passenger pigeon is, quickly Google it. It's a phenomenal and gorgeous bird. It was once the most abundant bird on the planet, numbering anywhere from 3 to 5 billion in the year 1850 to 1860s. But it's extinct today. From the 1860s to the 1890s, it was harvested at a commercial scale. The Industrial Revolution just came down on this bird really, really hard. And if you weren't around in, say, the year 1875, which I believe none of us were, I would hope.
Right.
Then, in the eastern U.S., most of the United States, what people don't realize is that at that time period, farms and ranches were not as established yet to provide food for the rapidly growing urban populations. And the commercial markets that provided food, like actual food companies, they employed hunters and trappers to go out and harvest wild game. And passenger pigeons would flock in these giant flocks of billions and billions of birds in one area. And so they were really easy to get thousands to millions to tens of millions all at once. And they were a staple food for about 30 years in the United States. And that harvest drove them to utter collapse by the 1880s. There were only tens of thousands of them left. By the 1890s, there were only hundreds of them left. And in 1902, the last one we know of was shot in the wild. A few stragglers at the Cincinnati Zoo hung on for a while, but they never bred. They were probably just from too few individuals, maybe a little inbred. And the last one, Martha, died September 1st, 1914. And you can actually go to Cincinnati Zoo today. They still have the aviary where she lived. There's an exhibit on passenger pigeons inside now.
Oh, cool.
It's one of two structures actually left from the 1800s in the entire zoo.
Whoa.
So it's a very historic place.
That's awesome. Gall with $25. Thank you so much. So our first chat question here, and it's one that I was going to ask anyway. Guy asked, so that's the passenger pigeon. What other species does Revive and Restore work with, or what are they working on bringing back?
Well, I am so happy someone asked that, because in the 8 years now that Revive and Restore has been operating, the last 3 have seen so much expansion for us. While I lead the passenger pigeon project, and I'm one of the world experts on that bird. There's only like 5 of us, by the way.
Cool.
And that's what started us, but de-extinction and extinct species are now just a small portion of the entire array of things that we do. And so this is the one cool thing I always want to point out, is that all of the technology you need to be able to sequence an extinct species DNA and edit it back into living cell lines and make embryos and hatch birds and recreate an extinct ecotype is what we call it. I can get into the terminology later, but everything you do for that, I mean, at every step of the way, these technologies could drastically help living species today. And so our first project away from extinct species was the black-footed ferret, which just started laboratory work this year. So it's been about 6 years of designing where we were going, and where that project is this adorable ferret. Definitely Google it and support them too. Supporting us also is going to save this ferret. Black-footed ferrets were actually thought to be extinct twice. That's how rare they became.
Wow.
And in 1981, a ranch dog dropped one on someone's porch in Meeteetse, Wyoming, and the rancher looked at it and thought, oh, gall, another dead animal the dog brought home, threw it over the fence. But his wife, she thought that was some strange looking animal, and she thought they should save it. So she brought it to a local taxidermist, and the guy lost his mind and said, do you realize this is the black-footed ferret? Everyone thinks these are extinct.
Oh my gosh.
And people came out.
I've never heard that. That's crazy.
They found the very last living black-footed ferrets out in this area of Wyoming. and they went in and captured as many as they could to bring into captivity before disease wiped them out. And since then, from only 18 ferrets, which actually there were a couple family groups related to each other, so from 7 non-related ferrets, they've bred nearly 10,000 ferrets in captivity. They've released them to 30 different sites between Canada, the United States, and Mexico. But the diseases that were causing them to go extinct in the first place are still a major threat on the landscape. And so there's a very effective vaccine which works, but for every ferret born in the wild, it's very difficult to go out and capture them and vaccinate them, then go back 6 weeks later and give them a booster shot. So those ferrets born in the wild have no protection from this disease. It's plague. It's the same Black Death that killed off millions of people hundreds of years ago in Europe. It got here to San Francisco in the 1840s on a ship from Asia, and it spread across the United States, and these native North American ferrets have no resistance to this disease whatsoever. And we believe that gene editing can basically take what the vaccine is triggering in these ferrets, and we should be able to edit in a way to where that's happening from birth, and it's inherited from one generation to another. And so we're starting some research actually in mice to explore whether or not that's possible. We're also working with cells from San Diego Zoo that were frozen 30 years ago that could bring back some genetic diversity to that species by cloning those cells. And so we're working on cloning to help endangered species like the black-footed ferret, gene editing for disease. Now we're working on I should get ahead of myself. We now have the Catalyst Science Fund, which is an explicit fund that we're able to work as a partner with labs that are doing things that no one else would fund because they're too risky. They might fail.
Okay.
So National Science Foundation, when they give $100,000 to a team that's going to explore some radical new technology, they want to know it's going to probably work, that they're going to not be wasting their money. Well, we're a small startup and we want to change the world, so we're a little more willing to take that bet. We now have 5 programs, 2 that we incubated with the black-footed ferret and birds, which I'd love to talk about more. But now we've got multiple programs from other partners that applied to us that we worked with that are Steve Palumbi at the Stanford lab, who's now using pharmaceutical drugs to simulate stress on coral reefs so that they can study how coral genetics respond to stress. If anyone knows anything about coral reefs right now, climate change is a major, major problem for them with major bleaching events.
Yeah,
and
we just talked about that like a couple days ago. I was in Belize recently and we did some diving and saw some coral, so we talked about it a little bit on stream. But
yeah, I mean, if anyone can get out to those areas and dive and see, living corals, it changes your life. And then when you see the fact that, half of the Great Barrier Reef has died off, that so many of these corals are dying off, that they're moving away from the equators because they can't handle the stress of the acidity and the heat in the ocean that's weird, that, we're pumping out there. That's where work like Steve Palumbi's becomes so important at figuring out ways that we might be able to, identify the genes involved in that and start to make some changes to those so that they can handle heat, because there are corals that live in tropical waters versus those that live in temperate waters, and we should be able to figure out ways to help adapt all of them to the changes they're experiencing. We have another program that's working on trying to culture elephant herpes virus.
I didn't know about that one.
It's a virus that kills off a lot of juvenile Asian elephants in captivity and the wild. And no one can come up with a vaccine for it because they can't grow it in culture. So we're kind of taking that first step to try and make that workable in the labs. We're sponsoring work at UC Davis on in vivo gene editing, the ability to maybe do gene edits in an organism without having to culture cells and create embryos and do the whole rigmarole of reproductive technology, which could really be amazing for combating invasive species or adapting some of these species to changes. So we're doing a lot with innovating science now for ecosystems and endangered species.
Yeah, that's awesome.
We did the and I would direct people to our Ocean Genomics Horizon scan, because we're now funding 3 different projects, additional projects on coral reefs. And we now have a fund to be able to start sequencing genomes of marine organisms for conservation purposes. But in the realm of the extinct species, we have passenger pigeon, heath hen, and the big media getter is always woolly mammoth.
Right.
George Church at Harvard is working on the woolly mammoth, and but it's really only those 3 extinct species, and all those other programs that are more and more programs with endangered species. Yeah.
Okay, awesome. Wow, you guys have a lot going on. That's so cool. So we said yes, heath hen. Okay, and then what else? I also have horseshoe crab here.
Oh yeah, I missed that. That's kind of an interesting one for us. So we're at Revive and Restore. We started with these in-house projects like passenger pigeon, heath hen, mammoth, black-footed ferret, where no one else was really steering those projects forward. So we took the reins and said, okay, we're going to make this happen. And as I said, we then developed the Catalyst Science Fund so we could start being a funding partner to enable other labs to really start working these things forward. But one of the other things we do is actually just switching the paradigm. We're bringing together biotechnologists with conservationists, 2 camps that rarely end up getting the chance to talk to each other, holding these workshops to really make them cross that bridge, and getting biotechnologists to think, what could we do for conservation? Getting conservationists to think of, well, if I had this, here's what I would do with it. And in bringing people together, we start learning stories. And the story of the horseshoe crab is such a phenomenal one, where we didn't have to develop the technology at all. The technology was invented more than 25 years ago, but just hasn't been adopted. And so that brought on like this 4th role for Revive and Restore, where we could see technologies that exist and start working as an advocate and a lobbyist to really get people to adopt those. So the story of the horseshoe crab is that anyone who's ever taken a drug in their life owes, and it has saved their life, owes their life to a horseshoe crab. And here's why. At every laboratory where pharmaceutical drugs are made, they have to test the water systems and the drugs and other aspects of the laboratories to make sure there are no contaminants of particular bacteria that could end up killing people.
Right.
And the best way they've come up with to identify these is using compounds in horseshoe crab blood.
Okay.
So every year, along the east coast of the United States and China, the places where horseshoe crabs live, thousands and tens of thousands of horseshoe crabs are captured and then milked for their blood, bled out a little bit.
How does that process work?
I'm not really sure. I think if you see the photos, they kind of fold them. Horseshoe crabs, they look like a horseshoe. They got a big
we have a picture on the screen right now.
Awesome. So they fold them, and then they puncture a little vein, I guess, or an artery, and then they release them. And while for years people thought releasing them, they would be okay, the populations have started to decline. And not only is this bad for the horseshoe crab, but horseshoe crabs, when they come and they spawn on the beach and they lay their eggs are a major food source for migrating birds. There are birds migrating from the Arctic to South America that purposefully time their migrations to hit the spawning events so that they have food to get there. And so it's this cascade of, you start losing horseshoe crabs, you start impacting the birds. Those birds are flying from the Arctic to South America, so that's impacting the species they interact on those very different places of the globe. So it has this huge reach. And back in the 90s, people used genetic engineering to figure out, one, they were sequencing DNA from horseshoe crabs. They figured out which protein in its blood makes that test possible. They found the gene that makes that protein. They were able to splice that gene into a bacteria. And the same way that bioplastics are made and insulin is made, they're able to grow lots of bacteria and basically extract out that protein from the horseshoe crab gene. And so there's this, it's called recombinant factor C.
Okay.
And yeah, it exists, and it's been on, available for 20 some years, but the pharmaceutical industry has not really adopted it, and there were some political reasons getting in the way, and really, so Revive and Restore took it on ourselves to start lobbying. Ryan Phelan and our former conservation director, Tom Maloney, actually put together a review paper of all the studies that have investigated the efficacy of this recombinant factor C, and said, it is actually as good, actually sometimes better than horseshoe crab blood.
Cool.
And yeah, they had a landmark meeting with the Audubon Society in New Jersey and the First Lady of New Jersey, where there's a major fishery for horseshoe crabs, to start calling for this, this synthetic alternative to be adopted and become a mainstay of the industry. Because even if pharmaceutical labs switch to this just for testing their water, it will reduce harvest rates by 90%.
Wow, that's amazing.
And yeah, it's really cool. But so it's not a technology we developed, but it's definitely a technology out there that's it's probably a premier example of how biotech can save an entire ecosystem. But you got to push through the red tape to get people to realize that it's there and that it's usable.
Yeah. So this photo on the screen, guys, is I guess what the bloodletting looks like. So that's what we're talking about right now. So we're at $71 in donations here, and it looks like your donations are going to a variety of projects, which is awesome. We love stuff like that. So if you wouldn't mind, let's get into the nitty-gritty of the passenger pigeon stuff. So how does de-extinction work? Where do you start? Walk us through it.
Well, I'll give you a kind of, the simple conversation version, but for anyone who's really interested in delving into the topic and going in detail, they can look up a paper I wrote that's open source, because that's how everything should be.
Nice.
It's simply called De-extinction. So you Google de-extinction Ben Novak, and then it's in the journal Genes. And it's a really long paper that explains how it's done, what our limitations are, the considerations for how we do this. And in general, what we always tell people is if we were simplifying this down to its most base element, you're taking DNA from the extinct passenger pigeon and sequencing it, getting its whole genome. And that's something that we did with the UCSC Paleogenomics Lab that was published back in 2017, that we had those genomes, and compare it to its living relative. In this case, the closest compatible relative is the band-tailed pigeon out on the West Coast. And my master's thesis is on this topic too. It's also out there if people look that one up. But basically, in these documents, band-tailed pigeon and the passenger pigeon, they're already 97% genetically identical. They're already extremely similar birds, eating the same types of foods, similar size, laying the same number of eggs. I mean, they're extremely similar. They're just different at a few critical kind of behaviors and traits. And those are the things we're zooming in the genome to try and figure out, like, what makes the passenger pigeon unique. And once we figure that stuff out, there's a lot of science involved in that. But once we figure that stuff out, we'll edit those traits into the genome of a living band-tailed pigeon. And this is where things get kind of particular for birds. If we were working with, say, a woolly mammoth or a type of rhinoceros, any kind of mammal, we would do the gene editing in, say, skin cells, and we would then use cloning to produce an embryo. We would take the gene-edited nucleus out of those skin cells in a petri dish and stick it into the egg cell of a surrogate mother. That would create the embryo. We would then implant that into a uterus, and 9 months later, a year later, however long the pregnancy is, you'd have your de-extinct mammoth or woolly rhino or whatever you're working on. In birds, because they lay eggs, cloning is almost impossible to do. No one's ever actually accomplished it. So people were, it was so difficult to try that people worked on a workaround, and it's called germline transmission.
Okay.
And basically, you're taking, rather than taking skin cells or any kind of cell, you're taking a very specific type of stem cell called the primordial germ cell, and you're doing your gene editing in those.
Okay.
And rather than creating an embryo and implanting that into a mother, what you actually do is you take the gene-edited primordial germ cells and implant those into a surrogate mother and a surrogate father.
Mm-hmm.
And then the mom and the dad breed, that produces your offspring.
Okay.
And so that's what we would do. We would take the primordial germ cells of the band-tailed pigeon, gene edit them to have the passenger pigeon traits. And for people who don't know gene editing, it's very different from older genetic engineering technologies. We're not talking about taking passenger pigeon genes and splicing them into the band-tailed pigeon genome. We're actually literally rewriting the band-tailed pigeon's code to match the code of the passenger pigeon.
Right.
So it's not like inserting new DNA. It's going through and going, oh, this T needs to be an A, and so it's far more precise. And that's, gene editing is the technology that actually makes this possible at all. And once we do all that editing, we have those cells. We'll put those probably not in a wild band-tailed pigeon. What we would probably do is implant those into surrogate mothers and fathers who are domestic pigeons, like the kind you see on the street all the time. Mm-hmm. Because they're really, really easy to work with in captivity. So we can provide them with really great welfare. And they also lay a lot more eggs every year, so we can produce a lot of offspring and kind of kickstart our new species that way. So this surrogate mom, the surrogate dad, which are called germline chimeras, then mm-hmm. breed. And if we've done our job right, when the egg hatches, out comes a new generation of passenger pigeons. And that's where the work becomes the mainstay of what conservation has been doing and becoming better at for the last 200 years. And that's breeding animals in captivity, conditioning them to live in the wild and releasing them back to the wild, and then, watching them kind of rediscover their environment and making sure everything goes all right. And there's immense amount of planning that goes into that.
Yeah.
And in the published paper, you'll find that rather than thinking of this in linear terms, you can think of it cyclically, because once we sequenced genomes, we started learning things about the species' population history that were telling us about their ecology and what things we would need to know about restoring them back to their habitats. And knowing what we know about their paleoecology and their behavior from historical records, those things kind of tell us what are the traits we're really looking for in the genome. So each phase of this actually kind of has some foundation research going all the time, kind of feeding into each other. And it becomes this kind of beautiful mosaic painting of research and work, where there's these new biotechnology steps that need to be innovated, and then these tried and true conservation steps that come in. I think what people out in your area would probably be most familiar with for conservation of this type would be the California condor. Mm-hmm. So back in the 80s, the California condor population crashed to about, I think it was 24 individuals.
Yeah. They
brought them into captivity and bred them, and the first ones they released to the wild all died, or they had to go out and capture them before they died and bring them back into captivity, because while they were in captivity, a lot of their environment had been developed, and when they were releasing them, they were landing on power lines. Mm-hmm. And it's such a huge bird. It's the largest bird in North America, that it would touch 2 power lines, electrocute itself, and die. So they had to start adapting them to this new environment. They had to start teaching them and training them. And once they started training them in captivity, don't sit on the power lines. When they were released to the wild, they avoided that. They would teach that to their offspring. The Condor program is a great example of a decades-long program in which they've figured out a lot of kinks to be able to breed and build up the numbers of California condors and get them out into the wild. And they've hit some great milestones after 40 years of work. They've I think last year or the year before, they started having more chicks in the wild than in captivity.
Wow, that's awesome.
And they're gearing up to reintroduce them into new areas of their former habitat, places they haven't been in centuries. And the more people look into this, the more they'll find examples of living species that have been saved by this type of work that are exact parallels of what we're doing in de-extinction. And those examples really answer a lot of questions. Like, one of the questions we get a lot are, well, it's been 120 years since the passenger pigeon's been in the wild. How is it going to go back to this environment? Hasn't it changed a great deal?
Right.
But actually, people have been reintroducing species to areas they've been gone from for centuries, sometimes millennia. Quite often, out in the East Coast, we have populations of elk in Pennsylvania and Tennessee that were, elk had gone extinct in the 1800s. It was nearly 100 years later that they brought them back. Turkey in New England, like wild turkey, which I've talked to lots of people up in Boston, and they think turkey are just like have always been there and are just so prolific. But turkey were gone from all of New England for 160 years.
I did not know that.
It was just 30 to 40 years ago that people started successfully putting turkey back.
Wow.
And turkey that live on the West Coast, actually in California, the last time turkey lived in California was 10,000 years ago. And people put them back into California in the last 100 years.
I didn't know that either. That's crazy.
Yeah. There's actually...
Everywhere.
Yeah, I think this is one of the coolest things about interventions in nature is we have generations of people now that are separated from this major historic event in the 1970s when the environmental movement and young people and old people alike just rallied to have the Endangered Species Act signed. And it was ratified by Congress in this like amazing show of partisan unity that we never get anymore. And you fast forward almost 50 years into the future, and you have generations growing up that were born in a world that protected wildlife and cared about wildlife, in a world where things like hunting were turned into conservation tools rather than slaughtering species. And so people really don't know a lot of what conservation has done over the last century. And they don't know that, when they go out to this wilderness area that sparks kind of transcendental feelings in their heart and their minds, that nature is not separate from human beings, and it really never has been. Nature has been pedicured and manicured and gardened by conservationists who are restoring it all the time from the mistakes from our past. And the one also awesome thing to remember is, if you think that there's somewhere you can go, especially here in the United States or really anywhere in the world, you can go to some national park and think, oh, this is what it was like before humans tampered with this area. Don't forget that when Europeans found North America, there were 100 million people already living here with cities and trade routes and agriculture. People who were shaping the landscape by doing controlled burns on prairies and in forests. The First Nations people of North America were shaping this continent for nearly 18,000 years. And so human beings are we're a part of nature, and modern conservation kind of keeps us part of that. And that's kind of the coolest thing I think about our work is, now that we're in the age of biotechnology, where genomes are, genomic science and gene editing are revolutionizing biomedicine and starting to change agriculture, it's like, let's bring that into conservation too. We've been intervening and restoring and helping nature now for over a century with every tool we could innovate. These are new tools that can just propel us into a new realm of success, and that's our nexus. And I definitely got off topic from passenger pigeons there.
No, that's
but
fine.
I think that's where this idea of putting passenger pigeons back in the forest goes to. because there's a very important reason why we would want passenger pigeons back, these giant flocks of 1 billion birds back in the forest. And that's because today, 120 years after they've disappeared from the wild, what few people also know is that we actually have more forest in the eastern United States than we have in 100 years. Starting in the 1920s, as agriculture shifted towards the west, abandoned agricultural plots, abandoned mining plots that got tapped out, started becoming reforested. So there are places in the eastern US now that have as much forest today as they had 200 and 300 years ago. And so we have all this forest, and yet the species that we think of as being integral to those forests, oak trees and sparrows of all kinds, and things like cottontails in New England, like all these very eastern iconic species, despite having more habitat, are declining. And that's because the habitat is not the right kind of habitat. It turns out that eastern forests, for so many hundreds of thousands to millions of years, were being shaped by passenger pigeons. They came into the area, and they just caused devastation, just absolute devastation. They overcrowded branches, breaking branches down, and 1 billion birds in one spot, tightly packed together. I mean, it produced a lot of crap. So they were pooping on the forest floor, snuffing out the life on the forest floor. And afterwards, you would have thought this is the worst thing nature could ever have done to it. But that guano enriches the soil and changes the biochemistry and clears out the understory so that new seeds, they're all starting at the same playing field. And now there's a new patch of habitat for regeneration to start. And those broken branches are letting sunlight come down into that forest floor. And so just like a forest fire rejuvenates a forest, that's what the pigeons were doing, and they were doing it everywhere all the time, moving from one place to another, keeping this dynamic cycle going and creating a mosaic, heterogeneous habitat system. And every major dominant plant, every major animal that lived in that ecosystem, from the Mississippi all the way to the eastern seaboard, were thriving off of that constant turnover. And today we have more forest, but we don't have turnover. And so it's all closed canopy. It's all shifting to being dominated by just a few of those hundreds of species. And all those species that need that turnover are in decline. And for 50 to 60 years, Forestry Service members have been trying to go out and create those turnover disturbances. But as humans, we just can't bring it to the type of scale that nature needs. Sure. And so the whole idea of getting back a bird like the passenger pigeon is that once their numbers really build up, they do the job for us, and we end up preventing the extinctions of dozens, if not hundreds, of other species, possibly helping carbon cycles, sinking carbon into the new growth, into the ground, sequestering it. There's a whole host of things. Some of the things I imagine sometimes are just the interesting economic opportunities for bird watching, but also organic farming. What a lot of people are aware that prior to nitrogen-based fertilizers being invented in the 1900s, guano, specifically bird guano, was shipped by the ton across the world from seabirds and other colonies where they could get a lot of it as fertilizer for agriculture. And there you go. If you have 1 billion passenger pigeons dumping a whole bunch of guano, you have a place where people can go out and harvest and have organic fertilizer for their crops. And of course, if we do the job really, really, controversial thing is that someday it might actually be a species that could be hunted. As I said earlier, hunting is now a conservation tool. In the 1800s, we were over-harvesting and over-killing species by the dozen. And the moment people started to realize that things were, not an inexhaustive resource, they changed the way they did that. People like Aldo Leopold came up with modern game management. And in over 100 years since the United States shifted to a game management system, there hasn't been a single game species that's gone extinct due to overkill. and that's a huge triumph
Yeah,
where
that's
they're
crazy.
able to, every license that a hunter buys basically is money that goes into monitoring that species and protecting habitat, and the revenue actually goes into other species that are not game species. And so, a huge sign of success would be if we could have enough passenger pigeons to where they could be hunted and then start generating revenue for even more conservation.
Yeah. Awesome. Okay, I'm gonna go through and thank a few donations that came in while we were having this conversation. So Loch Ness with $20, Spoon with $100. Thank
Wow.
you so much, Rocky with 50, and there were a bunch of other small donations and $10 donations, $25 donations that came in. Thank you guys so much. We're at $349.36 in donations. So thank you. One of these donations said, and it's backtracking a fair bit, but is asking about, I'm gonna say it wrong, chimeras.
Chimeras. Yeah, I
Chimera?
brought up the chimeras.
Yeah, would you mind defining that for us? American Eagle Foundation,
So,
$20. Thank you so much.
awesome. I wish I could send thank yous out to all of you individually. So chimeras, the term itself, a chimera is an organism that has cells from two different individuals into one unity. Every once in a while, there are chimeras born naturally. So when fraternal twins become one human being again, you get a chimera, and it's different from a hybrid. So when you have a hybrid, you have one species and another species breeding and producing a hybrid who has a mix of both genomes, both sets of DNA. Whereas a chimera is a mix of cells where the DNA itself is never mixing, but the cells have mixed together.
Okay.
And that's a lot of crazy, science to remember. But in what we're doing, when we take the gene edited cells of the living bandtail pigeon, which we're still several years away from getting to that step, we can get into that in a bit here. We take those cells from a bandtail pigeon, we implant them into the surrogate mother or father, and that would be a rock pigeon. And so that bird is now, it's every organ in its body, its heart, its brain, its muscles, its feathers are rock pigeon, but in its reproductive system, it's growing the new passenger pigeon cells.
Right. So we have a
And
picture
so that's
on
why
screen. We have a picture on screen right now of the world's first chimera. I keep saying that wrong. That Heather sent that over to me. So this
the
white-
world's first first one we made. So basically if you alter the genetics within just some tissues and not others, you've created a chimera. Any organism in which some of the cells have different DNA than the others is a chimera. And chimeras are really essential steps in genetic engineering, because we get some of their reproductive system engineered, and then those chimeras who have normal bodies but have genetically engineered cells in their testicles or their ovary, they can produce the genetically engineered offspring. And over the last 2 years, I was working at a laboratory in Australia, the CSIRO Australian Animal Health Laboratory, who are doing some groundbreaking work on everything from the environment to agriculture. And we were trying to produce the world's first genetically engineered pigeon as proof of concept. And there were several layers to this work, but the main thing is that pigeons just they're very different to handle than, say, a chicken. So almost all genetic engineering in birds to date happens with chickens or quail.
That's right. The first time that I had heard about it was Stuart Brand's TED Talk from forever ago with the chickens and falcons.
Yeah. And moving to pigeons just requires a whole lot of innovation, not in the high tech end, not in the biotechnology end, but just in breeding and handling the birds, how you take an egg and open it up and inject your new cells. And so we have to first get the reproductive system genetically engineered and produce the germline chimera, and then hopefully that chimera, when it sexually matures, will lay an egg that is the genetically engineered bird we wanted. We bred and raised 24 pigeon germline chimeras, the world's first 24. We're the first lab ever to successfully do it. Other labs have tried. And sadly, in that first round of experiments, the chimerism was too poor to produce genetically engineered offspring. And when I say that, what I mean is that if you took a semen sample from that male bird, that world's first germline chimera, only 1 in 100,000 of the sperm cells was genetically engineered.
Oh.
So that guy would have to have 100,000 offspring for us to get one genetically engineered chick.
Mm-hmm.
And that's impossible in a pigeon's lifetime.
Right.
Even though some people might challenge me on that with how many pigeons are in the world, it is impossible.
Okay.
And so, we really need something more in the line 1 in 100
Mm-hmm.
or 1 in 10. 1 in 2 would be phenomenal.
Mm-hmm.
And so, but the fact that we were actually able to produce the chimeras has proven that we've been able to work out the system to where we can create genetically engineered or gene edited pigeons. Now we just need to innovate a few extra steps to make that a better process. And there's a whole host of things we're doing to advance the field of avian science so that we can start making headway there. I mean, I've talked a lot about passive pigeons and a bit about how we would recreate them and the decades-long process of, what it would take to restore them to the wild. But, just figuring out what genes make what traits requires a lot of work. And if we were working in mice, it would be super easy. We would do gene editing in a single-celled embryo, and then 3 weeks later, we'd have a baby mouse, and we'd go, oh, wow, look, when you disrupt gene A, it's albino, so clearly gene A makes pigment. And if you disrupt gene B, he has no heart or something crazy like that. Of course, that's crazy.
That'd be unfortunate.
But, that'd be unfortunate. That embryo would not live. But this is basically how people figure out how genes work. They work with embryos and they disrupt genes, and then they look at what goes haywire and go, okay, this gene is vital to that pathway. With gene editing, now we can do things way better where we can change mutations, and rather than disrupt the gene, we can look at what different variations of the gene do. And so it improves the whole field and removes a lot of the kind of the ugliness to it. And for birds, the problem is we just don't have really efficient ways of doing genetic engineering or gene editing in birds. To do the same gene editing test in a chicken and a mouse,
Mm-hmm.
you could you could measure a trait in a mouse within 2 months, 2 to 4 months. And in a chicken, it's going to take you nearly 2 years.
Oh, why is that?
Well, that's because of making these germline chimeras is first. So in the mouse, you can do gene editing in an embryonic stage, and the mouse that is born is gene edited right away. Whereas in a chicken, you have to work on editing the cells in that chimera. That chimera has to take 6 months, 4 to 6 months to sexually mature. And then you have to breed them to get the gene edited offspring you need.
Mm-hmm.
And if only 1 in 10 or 1 in 100 sperm are carrying this trait, then you've got to breed up, 10, 100, sometimes 1,000 chickens to get your one genetically engineered bird to see your trait.
Right.
And so it's just way slower. It takes a lot more intensive resources. And part of what we were trying to do in Australia with the Pigeon Project was develop a strain of birds that would rapidly accelerate this process to the same levels that you can do in the lab mouse.
Mm-hmm.
And that's something that we're going to reattempt with a newer technique. Well, not a newer, sorry, a different technique here in the United States. Now that I've moved back to the States, I came back here because my wife and I had twins, and they were very difficult to raise in Australia, so many thousands of miles away from family that could help.
Mm-hmm.
I jokingly say that my twins are Revive and Restore's first clones.
Oh, that's
Um,
cute.
And now that we're back in the States, we'd like to, kick off phase 2 of this work here in the States. We're actually going to shift to working with quail.
Okay.
And this is because we want to actually test to see if this system's going to work before we started again with the pigeons. In Australia, we were kind of going for gold right away. But we believe we can test this out in quail in about a year. And it's a way of making a transgenic quail using this existing biotech that makes genetically engineered birds and doing 2 generations of birds the long and slow way to eventually develop a strain of birds that carry a gene that will actually make it easier to do gene editing in those birds so that we can maybe do it at the same rapid rate as we do in mice. And we can start really accelerating the pace of discovery for birds, so we can figure out what genes are involved with disease resistance for wild birds that are facing exotic diseases, and what genes are involved with climate adaptation for birds that might be struggling with climate change. We can sequence historic birds of endangered birds, like those California condors. We could go back to museum specimens from 100 years ago and find out what was the diversity like before that population almost went extinct. And we could actually edit back in some of that diversity and bring back some of that viability that's been lost.
Okay.
You could think of that as de-extinction at the genetic level. But there's all sorts of things we could do more rapidly if we could start figuring out how those genes work and have it go from being 2 years to just a few months. And so, that's actually what we hope to put some of the money that people are donating into today is making that major breakthrough. And if it works with the quail, we can move on to the chickens, to pigeons, to parrots, and start really advancing the laboratory work so that we can make discoveries that'll help endangered species of birds all over the world.
That's so cool. Speaking of donations, again, lots have come through. Rocky with another 50, Danny with 71, and Gall with a handful of donations. Everybody, Botox, thank you guys so much. We're at $578, so we surpassed that $500 goal a few minutes ago. If you wouldn't mind, I'd like to get into some of the viewer questions because they have a lot of really good ones. I think we've covered some of them. Oreo with $88. Thank you so much.
What amazing and specific numbers. Thank you guys so much. Someone just donate $3.14 for fun, like give us
That's
a piece
nice
of pie.
Number. He actually donated this amount because now our amount is at $666.66. So well done. I guess
there's an old blog post that someone wrote years ago where and they found the best picture of me. I have a mustache with curls and a little patch here, and they called me an agent of Satan.
Wow.
Myself and Stuart Brand, that we are agents of Satan that are messing with nature. And it was this I don't know if it was a joke or serious, but it was this like Christian Science blog that I think they were poking fun at people. But it was the day Leonard Nimoy died, and they said that he was going to hell and all kinds of crazy stuff. But it was so great to see myself as an agent of Satan in such a comic and satirical fashion. And so I'm glad we hit that donation of 666.
Yeah, well, now we're at 671, so we're okay.
We've moved into cleaner territory.
Yeah, thank you guys. So the first question, this was asked a while back. We have a couple on the Tasmanian tigers. And Tofa asked about the reintroduction of a Tasmanian tiger and how that would affect the ecosystem that they'd be reintroduced in. So that's Tofa's question.
Yeah. Well, so number one, the Tasmanian tiger, or the thylacine, was the extinct species that really got me into this. When I was 11, 12 years old, originally I wanted to be a paleontologist and work on dinosaurs, but then I became really, really into modern biology and animals. I was really wanted to be a conservationist, but I loved extinction. So it was when I discovered that there's so many species that have gone extinct in recent times that I could kind of maybe merge the two together.
Mm-hmm.
And the Tasmanian tiger was like the first one
Yeah.
that made me aware of that. And it's such a beautiful animal, and its extinction is so sad. That kind of reintroduction, if you could recreate something like the thylacine, which would be very, very difficult, by the way.
Mm-hmm.
Like I said earlier, the bandtail pigeon and the passenger pigeon are already extremely similar.
Mm-hmm.
A woolly mammoth and an Asian elephant are so identical that I know paleontologists that have found bones in the Midwest and said they were mastodons or mammoths, when in fact they were actually circus elephants that had been buried in
Wow.
someone's backyard from the 1800s.
That's crazy. I would not have guessed that.
They're
that they were
that close
similar.
to each other, that they even fool the experts. And we're looking at the tiny minor differences that would make a bandtail pigeon hyper social and flock in these big numbers so that it creates this disruption of the forest. We're looking at tiny differences that make an elephant grow woolly fur and be able to live in a cold climate. Whereas the Tasmanian tiger, it's drastically different from all of its living relatives. And so it's going to take a lot of hard work and science to get it back. I don't think it's impossible. I think we'll get there. And I think that, one, having gotten the chance to visit Tasmania when I lived down in Australia, over a quarter of Tasmania has been set aside as conservation preserves and national parks and national lands. So it's got a whole lot of great habitat. It has almost all of its kind of prey species still. And when you reintroduce a predator like that, you're putting back a very vital role in an ecosystem. Apex predators are, we actually don't even appreciate how important they are in ecosystems until they're gone, and then we would put them back. The best example we have here in the United States are wolves in Yellowstone National Park. And no matter the controversies surrounding the wolves on, say, ranching and economics, in the park itself, wolves have been a huge beneficial impact on the ecosystem. So wolves were gone from Yellowstone National Park for 75 years before they were reintroduced. And the elk population in that time really boomed up and became very large. And what they ate, the vegetation they ate, then became kind of overgrazed. And so there's the huge ecological changes when you bring out that predator. And by putting it back, they've reduced the elk herds, which has allowed tree saplings to start coming back up. And so because there's been more woods coming back up, it's actually changed the shape of streams and how the soil is eroding. And with the new woody vegetation, the beaver, which was also extinct in Yellowstone National Park, came back on its own.
Oh.
And has started creating beaver dams, which is now habitat for breeding amphibians and little fish and birds. And the kills from wolves are a boon to mesopredators scavenging. And it's created so much change to the park for the better, that I would suspect that you'd have something very similar with the thylacine, where you'd bring thylacines back, and just that small role they play in taking out some of the kangaroos or the wombats and some of the other major herbivores would have an impact on the vegetation, which would be, a cascade of events to the insectivores and the pollinators and all of the species that then result from that.
Yeah.
That's really what this comes down to, is transformation. I mean, the world is never static. It's always changing. And some people might ask, like, well, why do this at all? And it's really about going from negative states to positive ones. And that while the world is always changing, you can never go backwards. It's not like putting back passenger pigeons or wolves after 75 or 100 years goes back to some pristine earlier time. No, it's a new world. But we're converting what was a degrading negative state into a positive one that helps other species, increases biodiversity, bioproductivity, and bioabundance. Those are the three big B's that we care about, and what we're trying to help with that fourth big B, biotechnology.
All right.
More questions?
Yeah, absolutely. Let's it's similar. I mean, it's kind of going, oh, well, actually this one's highlighted. So, Michigan asked, so you can only bring back species if there's a living species that's similar enough to the one you're bringing back.
For now.
Okay.
I go into this a little bit in the paper published. The truth is we know so little about genetics yet that we're forced to really look at something that's very similar and make small changes. We cannot yet synthesize an entire genome of a bird or a mammal or a reptile or a plant. Some plant genomes are way too big to even work with. So like a redwood tree genome is 10 times the size of a human genome.
Really?
Whereas a bird genome is 1/3 the size of a human genome. So I mean, it's just we can't recreate and just write an entire genome and package it into a cell. And someday when we can do that, when we can just print out a genome in real life, package it into a cell, we'll be able to possibly bring back just about any extinct species that we can sequence its DNA. But one of the problems there is getting the DNA. So that's what I'm trained in. My laboratory skills are in ancient DNA prep, getting DNA from old museum specimens. So the next time you go to a natural history museum, you see all those stuffed dead animals, think you could take any bit of tissue from that and get its entire genome. It is difficult. Once an animal dies, its DNA starts to degrade into tiny little bits. And so you really can't put it back together in perfect order again. So we rely on aligning those tiny fragments to where they match on a living species genome. And so we actually need that living species genome just in digital data form to be able to start making sense of these fragmented and broken bits of DNA from extinct creatures. And the more unique an extinct species is, the more different its DNA is from a living species, the less likely we can match that up and put that back together. So there are some phenomenal extinct species that are so unique that we really can't get their entire genome right now. And there are people working on ways to maybe work around that, but even then, even if we can work around that problem, DNA does not last more than a few 100,000 years under the best of conditions in nature. So the world's oldest genome right now comes from an ancient horse bone. And it turns out it's just a regular horse, but it's a horse bone that's 700,000 years old. Jeez. And the only reason we were able to get DNA from it from that old, that long ago, is because it was in permafrost in the Arctic. And so it's been frozen for 700,000 years. You think of the dodo bird from the island of Mauritius, this, one of the probably world's most favorite extinct species. Who wouldn't want to see a dodo bird back? I personally worked on dozens of dodo bird fossils and couldn't get more than a single piece of DNA because they're tropical. That hot environment is just obliterating DNA. And we do have a dodo bird genome. The scientific community has finally got one from a single bone that actually was from a dodo that was shipped back to Europe.
Wow.
And died on the way. And so it was preserved in Europe where it was cool. There has not been a single dodo bone from Mauritius that has produced dodo DNA. And so tropical animals are more difficult, and animals more than 1 million years ago are more difficult. So, I mean, like, there's a lot of stuff going against things like you're never going to see a dinosaur again with what we're doing. Now, there might be a future in 50 years or a century from now or 2 centuries from now in which we understand genetics so well that we can create traits from scratch without knowing what gene makes them, where we can go, all right, I want to turn a bird into a dinosaur. And for all the nerds out there watching who are paleo people, I know birds are dinosaurs. So we're saying, for the sake of this thing here, we're going to turn a chicken into a Mesozoic dinosaur.
Right.
You could do that, but it's going to take tweaking genes and development, and what you'll get is something that looks like an ancient dinosaur, but it has absolutely none of the actual genetics from that ancient dinosaur. when we put mutations that we find in a passenger pigeon genome into a living bandtail pigeon genome, that new living animal actually has the genetics of an extinct species. And so we might be able to make some really unique creatures someday, but it just doesn't have the same essence or the same heart or legacy as what we're trying to do right now.
Yeah, got it. That's cool. So thank you, Mr. Take, for the donation. Courtney for the donation. We're $716 in donations. Thank you, everybody that's donated. I know I can't read all your names, but thank you. That reminds me, there was a question on donation from American Eagle Foundation. Connor asked, once the passenger pigeons released into the wild, how many years would it take to see billions of pigeons?
Well, that's a big question. And in fact, if there's any, science majors out there, people who do ecological modeling, stuff like that, these are questions that people can start to figure out on their own. If you understand the birth rate of the birds and how you might be able to work with them in captivity. This is actually some of the work we've done. We did some early tests on bandtail pigeon breeding in captivity to see just how many offspring you could possibly produce in a year. And, I think we can get to the tens of thousands in our lifetime, possibly hundreds of thousands in our lifetime. And a flock of 100,000 birds will probably start to make a small ecological dent, but we really need to get them up to the millions range. And that's probably something that'll happen, over the course of a century or two. These birds probably could get up to 1 billion in a few hundred years, but that would go a lot faster if we were supplementing the wild breeding with constantly releasing birds into the wild from captivity. We can produce probably 5 or 10 times more offspring in captivity to release to the wild than what birds would naturally be breeding in the wild. And so that's how we can get up to tens of thousands possibly in a couple decades. But that's going to take huge amounts of resources, and dedication from conservationists with multiple breeding facilities. And you just have to say, we as a society have to decide this is something we really want to see happen. We want the value of it to happen, that we care enough to see it through, and we can definitely accelerate it. But even without accelerating it, I have no doubt that hopefully someone will take up the legacy when I finally retire at the age of 98 and carry this forward. A really heartwarming story that came out just a week or two ago was the fact that a particular species of Galapagos tortoise, they did 40 years of captive breeding and releases and conservation to bring this species back from the brink of extinction. And they ended the program just a couple weeks ago because they hit success. They didn't need to do it anymore. And so they hit success. And so there is an endpoint where we can get to, and they had a lot of dedication, but it took 40 years. If that was at least 2 back-to-back careers.
Yeah.
And there are people out there that are still dedicated to other species that might be programs that last 100 years that are, you're looking at maybe 5 generations of directors. And so I think this comes back down to one of the major goals of our work is this long now view, is that we as a society, we've been around for a really long time. Modern human society has been around more than 10,000 years. And we are conscious enough now that we could stop being irresponsible and stop thinking short term and stop doing things that we know are going to have negative impacts in just a few years because they have a positive returns right now, and start thinking, what can we do today that changes the world for the better 100 years from now, 1,000 years from now, things that might have legacies that go 10,000 years. And we can always take note from societies that have lasted a long time, such as the First Nations people who had oral traditions that lasted for thousands of years and were passed on. You can look at the ancient artifacts of Egypt and Mesopotamia and look at the fact that human beings have been able to build institutions such as Oxford in England. It's over 1,000 years old. We can do things that last and leave a legacy for a long time. And I think that where we're at with revive and restore and this crazy notion of de-extinction is just that, is taking it to the next level of, we want to have lasting legacy and maintenance in nature that lasts a very long time, and really make a relationship between humans and nature that I see drifting apart, right? We have more people growing up in urban environments that very rarely get the chance to interact with wildlife and wilderness ecosystems. And, as I said earlier, all those ecosystems are already being worked with by people. There's no such thing as some pristine nature.
Mm-hmm.
And I think that's a beautiful thing, that we're always part of nature. No matter where we're at, no matter how far separated we think we are, we're always, as humans, part of nature. And biotechnology gives us like this new perspective, this new lens to be part of nature all the way down to the level of the gene. And that's like this just amazing, amazing power.
Yeah.
That we definitely at Revive and Restore want to see do amazing beneficial things to save species.
Okay.
And that is, I haven't introduced that yet. I mean, that's where the mission statement comes in, the genetic rescue for endangered and extinct species. That is what we call genetic rescue, the ability to go in, use the power of genetics to help species in nature in a century where it's facing problems that it's never faced before, with climate change, with global trade, spreading exotic diseases, with pollution, like it's never been at scales it's never been before, plastics building up in the ocean, all sorts of things that biotech is uniquely poised to bring new solutions to. And there are tons of young people out there innovating brand new ideas. And like everybody donating to us right now, we get to keep putting money into those innovative ideas for this next generation.
Yeah, cool. Oreo, thank you for the $61.77. That put us at $777.77.
See a pattern here. We're really
Yeah.
going for those unique numbers.
Good job, guys. Thank you. So a lot of these other questions that we've gotten here are pretty similar, and they all are about repercussions or pushback. So what is the pushback? What's the most common pushback that you guys get? How do you address that?
Well, this is such a common theme that Revive and Restore is actually hosting a unique workshop this June that I'm co-coordinating with our executive director, Ryan Phelan, in which every time we talk about intervening in nature, people want to know, like, well, what could the unintended consequences be? And what are the ramifications? Like, are you thinking this through?
Right.
And it comes from every angle, not just non-scientists in the public. It comes from conservation biologists. It comes from politicians. It comes from even people who work in conservation itself on the ground.
Right.
People are wondering, like, if you put passenger pigeons back in the wild, could it go wrong?
Yeah.
What are things that you would do to stop that? And those are things that are actually what we find is, the more we look into how people do this, how conservationists do it, how people have done it for decades, it's become a further and further refined technique where it's built in, where when people plan these types of things, they look at what is the ecosystem today. Let's go out and study it a bit, understand, we'll model the impacts, maybe do, and it's never the kind of thing where you take a whole bunch of animals, release them into the wild and go, let's see what happens.
Yeah.
It's an iterative process that takes years to decades, where people, like for the passenger pigeon specifically, what we would do is, one, we have a citizen science program that could greatly shape this. And we have to kind of rewrite the blog a little bit more. But if you jump on Revive and Restore's blog post, you'll find a citizen science blog where anyone in the Eastern United States could print out our form, and they could go out to their backyard, collect up a bunch of oak acorns, and preferably you would write down what kind of tree they came from: black oak, red oak, pin oak, whatnot. So the amateur botanist, is definitely someone we need involved. And you can cut little holes out of, say, the back of a notebook.
Okay.
Those holes are models of the passenger pigeon's mouth. And if you just throw the acorns through, say 100 acorns, throw them through and say which ones went through and which ones didn't. Say, out of 100, 60 went through model 1, 40 went through model 2. and 20 went through model 1, that kind of thing. That's going to give us the kind of data we need throughout the whole forest to measure what the potential impact would be from passenger pigeons consuming acorns. How many acorns are going to be left over for other animals that eat acorns? What is going to be the impact of a flock of 1 billion birds when they come in and they eat up all the acorns? Well, it turns out they don't eat all the acorns. They leave, they can only eat the smallest ones or the medium-sized ones. So it leaves these big ones for other species to eat. That's the kind of thing we need to figure out. These are all things we do in advance. And so, like I said, if people want to get involved, go to our website, send us data. We can make this happen.
Yeah, that's cool.
Responsibly. And there's other things we study too. We study the genome to understand how they would react to climate change, because based on how they lived in the past, all sorts of things we do to kind of make the program to where when we actually start releasing birds into the wild, we're doing it with a plan and a strategy. And first, we would actually release birds into what's called a soft release, a large enclosure that is a natural ecosystem surrounded by a natural ecosystem, but it's contained so that the birds can first get used to the climate, the weather, be exposed to the elements and hear other animal sounds. And then those birds can be released to the wild. And one, that allows us to know that the birds themselves are going to do well, but it also then prepares those birds for dealing with things in the wild. In the future, the GPS chips we can use are getting way smaller and way better. So all of our birds will be able to be GPS chipped to where someone sitting at a computer can actually watch the flocks move from one area to another live by satellite.
Cool.
And so we can be able to actually tell, are our birds flocking the right way? Are they possibly going somewhere that we don't want them to go? Like, there are going to be some potential issues that happen. And it's all about having mitigation strategies.
Okay.
The one thing people worry about, of course, is, well, what if there becomes too many passenger pigeons and they overrun us and they do something horrible? And my one thing to remind people is that people with muzzle loading shotguns killed off passenger pigeons in the 1800s. So we already know the recipe for controlling them. But yeah, that's a little cynical way of looking at it. But yeah, when wolves were put back in Yellowstone, when elk were put in Tennessee, when these interventions have been done, there's a lot of people planning what the unintended consequences would be, so that those programs don't have bad impacts on the environment. And it's actually a hugely not what you would call a hugely mature field, but it's a field getting better all the time. It's what I would say is it's scientific. It's very scientific. And right now I'm actually doing research to kind of quantify, like, over the last century, how many times have animals been released to the wild and how many times did it ever go wrong or do something bad? And it turns out that, things don't go bad very often, if at all. We're talking really, really tiny numbers, the kind of things that if you were doing anything else in the world, you wouldn't even look at. And there's tons of things people have done in nature that you would think could have huge repercussions, and they end up being beneficial, and sometimes even beneficial in ways that we don't intend. Really great example comes from my home state of North Dakota. There's an endangered orchid that lives now on just a few different protected areas of grassland called the Western Prairie Fringed Orchid. And in Montana, the neighboring state of Montana, and in Canada, just across the border, some USDA officials released an Asian hawk moth to control an invasive weed called leafy spurge. This is called biocontrol. It's done actually a lot. And so this was a new species introduced to the system to control this other new species that they didn't want there. So you have one invasive species doing something bad. You're bringing in another invasive species to try and control it. And in the past, 100 years ago, when people started doing this and they didn't really have the science behind it, yeah, some really bad stuff happened. You brought, like, cane toads in Australia. They brought those in to combat another foreign species, and then the cane toads didn't eat those at all, and they started causing problems for all other species. But now in the last century, this has become so refined that it's almost exclusively beneficial. We're talking at 99% of the time it works. And so they brought in these hawk moths. Well, these new hawk moths, then dispersed and started going other places. They moved into North Dakota, which was an unintended consequence. You know, that's not where they were released. But when they made it to the national grassland where this endangered orchid lives, not only did they start feeding on the invasive leafy spurge and controlling that weed, so that national grassland got that benefit, it's actually become the #1 pollinator of this endangered orchid.
Oh, interesting.
And those populations are stable today because this new introduced hawk moth is pollinating those flowers. And that was an unintended consequence. It's not something that anyone planned for, but it was totally beneficial and positive. And those are some of the crazy things that happen. And so, you know, when you hear that the world is going to hell in a handbasket, and you hear about all these species going extinct or declining, or invasive species having problems, I think the one key thing to keep in mind is that invasive species that are problems are almost exclusively accidental. Like rats on islands around the world are invasive, and they got there as stowaways on ships, you know, over the last 500 years. When people intentionally release a set of organisms to do a purpose, it's gotten to a degree of scientific quality that we don't end up having those giant ramifications. And so I have a great degree of confidence in this, but that's also because we're building that into the work. So those types of questions are built into the work from the beginning.
Yeah, so Sile asked about risk management. He said, what are some of the risk management strategies implemented to combat the unpredictability of the ecological effects? So your risk management is happens before the fact.
Well, you start planning it before the fact. So you start doing that modeling. You try to anticipate what those negative impacts would be, what your worst case scenario is, and then you come up with what would your mitigation strategy be. For passenger pigeons, you know, one of the risks is that they end up harming the species they're supposed to help. And the reason this could happen is, let's say, so the New England cottontail is a great example. It's critically endangered because it no longer has the types of early generation habitats it needs in forests. So we would bring back passenger pigeons to create that habitat. So in essence, it should save the New England cottontail. But the New England cottontail numbers are so fragmented today on such little patches of land that, you know, if you bring back passenger pigeons, a giant flock could come into the last little bit of forest where there's a few hundred New England cottontails. And when they come in and they do their damage, you know, they're creating a disturbance that could wipe out those rabbits, especially if those rabbits have no suitable habitat next door to run away to. And so you would end up creating this giant problem when you were really supposed to save them. And that's where tracking the movements of these animals comes in. If you can GPS track the birds, know where they're headed ahead of time, you can start with mitigation strategies such as sound, like putting out giant speakers to play frequencies to keep the animals away, or actually going in and moving the rabbits before the passenger pigeons get there to a safe area so that they can then go back the next year when that habitat is pristine for them. And then they can start really taking advantage of that. And that's the problem, is so many species are in precarious states that even the things that should help them, if not done in the proper sequence, could end up harming them. And so, sonic speakers to keep them away, possibly setting up some low-level hunting to thwart them away, with things like if you're worried about particular endangered types of plants that are rare, you can go out and put out particular types of netting and enclosures. Like, there's a lot of things that can happen on the ground to prevent a flock of passenger pigeons from creating damage when they come in, so that those species can really take advantage of the regeneration they bring. That's unique to passenger pigeons. For other species, there's, you know, all sorts of different ways you can manage these. Oftentimes, mitigation strategies have not needed to be employed But there have been conservation efforts in the past where they had mitigation strategies planned and in place, and they did end up having to resort to them, either to prevent the animals they released from dying off, or to prevent them from causing a problem for other animals they were trying to maintain as well, or plants.
All right. Guys, those are really good questions. Spoon tip $200. Spoon, you're insane. Thank you so much. So the last question I have here, I think it's the last question, is why are they called passenger pigeons? Is the passenger part significant?
For anyone wanting to really delve into passenger pigeons, you know what? Just a minute. I'll be back in 10 seconds.
No worries. And that question's from Remy. Spoon, you're nuts.
All right. These are the two books you want to try and track down.
Okay.
And one of these, I think, is reprinted on Amazon. So back in the 50s, Alan Shorger, the god of us passenger pigeon lovers, wrote this book, The Passenger Pigeon. It's Natural History and Extinction. And this has almost everything you'd want to know about the bird. It also set off a lot of misconceptions in the following decades, which my research is working on correcting. But it's mostly beautifully accurate. It's an amazing book.
Okay.
And then this new one by a friend of mine, another passenger pigeon expert, Joel Greenberg. And this really tells way more of the human side of the story, kind of the crazy characters that were involved in the passenger pigeon history. And both these books will give you kind of everything you'd want to know about them. And not necessarily, as I said, there's a few probably older misunderstandings from where the science was that we've now worked out with the genetics and the studies that we do, which I will share. But what you'll find from the books is that no one knows where the term passenger pigeon comes from. Some people believe that it's because they thought that the flocks were so dense that they were riding on top of each other like passengers. No matter how the term came about, I have a personal theory as to why it became popular. Because originally, in the 1500s, when the first accounts of passenger pigeons were noted, they were simply called wild pigeons. Later, they became known as the American wild pigeon. People called them wood pigeons. And I think, it's in the late 1700s to early 1800s that the term passenger pigeon really starts to take off. And I think it might actually be related to Americana and the Revolutionary War, the fact that America, the United States of America, became a country and were separating themselves from European culture. A lot of words and language changes were tweaked in the early stages of the American history. And I think the fact that passenger pigeon was a non-European name had to do with why it became so prevalent, that in the early days of the United States, people wanted American names for things. And no matter where it came from, it was the Americanized name for that bird, and it gained popularity. So that's my personal idea. But where the name comes from, not a clue.
All right.
If you do read those books, as I said, I'd give you some of the new information. One thing you might get from those books is that the passenger pigeon had this ideal habitat somewhere around the Great Lakes. That's not true. So they bred, the majority of records of where they were breeding is in this belt around the Great Lakes that Shorger calls the principal breeding area. But from research we're doing, that looks like that's just an object from historical bias. So it's like that's where most people were living at the time. So that's where most of the accounts come from. There are accounts of passenger pigeons breeding in Mississippi, in Louisiana, in Texas, in North Dakota, Minnesota. I mean, they have a home range, and basically every woodland of the Missouri system and the Mississippi system east, from Hudson Bay to Florida, is passenger pigeon habitat. There's no special area for passenger pigeons. Another thing that's always been up in the air is, and this is a big thing that lends support for the restoration we want to do, is people argued that passenger pigeons, in their billions, were a recent phenomenon created by Europeans. And this was popularized by Charles Mann in his book 1491. And it stems from some outdated research now that's been corrected several times. Was the idea that when Europeans got to North America and brought in new diseases that wiped out the First Nations people, that the passenger pigeon was able to explode because it was no longer competing with humans for food, and it was no longer being hunted by First Nations people. There's no support for that whatsoever. And in fact, none of the models and none of the data ever supported that. And our population genetics actually show that passenger pigeons were hugely abundant for 50,000 years, possibly as long as 1 million years. And so they predate humans even arriving in North America. So giant flocks of passenger pigeons were a natural mainstay of these ecosystems as for thousands and thousands and thousands of years and generations of every organism in the environment. And those are two of like the biggest misconceptions that you get from the literature sometimes, that the new research is showing are just rooted in misinterpretations of old historic bias.
Okay, nice. So that's again a lot of really good questions, you guys. Thank you to everybody that asked questions. We're at $1,111.11.
Wow, awesome!
Donations.
That's incredible.
That's a number. That's quite a number, guys. Thank you so much. Yeah, Maine with $60 and his first donation. Thank you so much. We really, really appreciate that. That's awesome. And Rocky with $100. So $1,211.
Thank you guys. Thank
Rocky,
you.
thank you so much. Brilliant. So that pretty much sums up all the questions that I've seen come through chat today, or that Saito has seen come through chat. Everybody, hearts in chat again for Saito, for compiling this doc for me and helping with my research and everything. Is there anything that you would like to talk about or share with us before we close up here?
Let's see. Well, we covered genetic rescue. We covered how de-extinction can help endangered species. As I said, some of those donations coming in can go to helping us advance research in birds. If you get on our website and look at the Catalyst Science Fund, you can see the projects we are pushing forward. One of those is work with birds. So the passenger pigeon project, the heath hen project, basically any work with an endangered bird, it all comes down to needing to advance that primordial germ cell research I talked about. So that germline transmission, that unique thing in birds, right now it's something that's only really possible to do effectively in domestic chickens. And when people try to really optimize it for other birds, like I said, when we first tried this stuff in pigeons, it was just so drastically low, we couldn't really get what we wanted. There's some steps that can be done to advance that. And one of those is culturing the germ cells in the laboratory, being able to grow those up. And that's really difficult to do for new birds that are not chickens. And we have a program that just got off the ground in 2019. Our Catalyst Science Fund has put $350,000 into a partnership with Texas A&M University to develop that reproductive technology for the first time in a wild bird. We're taking baby steps. So we're going from the chicken to a bird called the prairie chicken. Baby steps so that we can move to a wild bird that we think is very similar to the chicken, hopefully figure out some first bits there, and then as we get more funding coming in to expand the program, we'll move on to birds like the pigeons and the parrots and whatnot. And I'm really excited because this could be, in the next 2 years, we think we could have some of the first real breakthroughs with wild birds there. And I think all these technologies are absolutely accomplishable. It just requires continual support and innovation. People have done some absolutely astounding things in the past. When I was 14 years old, I had a Scientific American article that talked about a physics lab that had stopped light in a vacuum. They had managed to slow light down in a particle of atoms and then actually trap it in time and space in one spot and then let it go. And light in a vacuum, that's a constant. It's energy. The fact that humans could manipulate and harness light in such a way, it blew my mind, and it told me that there's really nothing we can't achieve if we really figure it out, and put ourselves to it. And all these biotechnology revolutions we need to make this happen, absolutely doable in our lifetime. It might take 5 to 10 years to get these innovations to begin thinking how to create a passenger pigeon, but we will absolutely do it with support from people like who are donating today. Spread the word. You can follow me on Twitter. It's at Ben J Novak 1. I haven't tweeted in a long time because I use Twitter usually to throw out updates on the research. And as I said, we're actually now transitioning that research from my work in Australia last year to here in the United States, hoping to kick that work back up in July. So every dollar coming in can start resuming that. And once we get some neat work updates on, those will be going on Twitter. We have a Facebook page for Revive and Restore, a Facebook page for the Great Passenger Pigeon Comeback, and of course our website. Revive and Restore is on Twitter as well, and our website's always a work in progress. You can see all the details we have about our different projects on it. Check out our annual reports for the major updates and the blog posts for new news. But the one really cool thing we did over the last couple of years that helped expand these projects, like I talked about earlier with coral reefs, was something called the Ocean Genomics Horizon Scan. And basically, Revive and Restore was granted funding to explore what biotechnology could do for oceans. And it was interviews with over 100 different scientists around the world, literature searches. It was the work of almost, I think, 20 different people. And it's this culminating document that confronts what are the 10 leading threats right now to ocean ecosystems and life, and what are the emerging biotechnologies that could help, what are existing biotechnologies that could be leveraged for this if they were adopted more. And then rather than just present a report on this, we decided at the main office with Ryan Phelan's leadership to really steer this into change. There are these what we call 10 big ideas, and we are fundraising for those 10 big ideas, where each one is an innovative project that already has a team of scientists ready to go. They've got everything figured out what they want to do. Money just has to come in to start, where each one is confronting a different major problem to ocean ecosystems, from advancing science for coral reefs to advancing the science for restoring islands by eradicating rodents on islands, looking at how that helps the entire ecosystem, to using environmental DNA monitoring to assess deep sea mining activities, so we know that those are actually not creating harm to the ocean, or if they are, how we can change those practices, all the way to new types of tracking pollution in ecosystems. It's an amazing assortment of ideas. And the Coral Project has actually already received partial funding, and the project to sequence ocean species genomes has received partial funding. So those two projects of those 10 big ideas are actually up and running this year. And that report, it's long, it's 170 some pages, and I'm sorry I couldn't catch every typo in it, neither could every other editor, but it's a huge resource of the type of people in the world that are doing this. So if there's a lot of college students out there interested in how they can get involved in this work, you go to those documents like that Ocean Genomics Horizon Scan, you go to the literature that's been published, and you go, who's working on this? Who's doing gene editing in corals? Who's doing reproductive technology in birds? And that gives you a lead of which university to go to, who to talk to, because this is an expanding field. There's room for lots of different disciplines in it, and every student could go to university for zoology, for paleogenomics, for genetic engineering and biotech, for conservation biology. There's a ton of different ways people could get education to work in this type of field. There's tons of ways that citizen scientists who don't have these science backgrounds can get involved. And for the social science people out there who are going into politics, those are the ones we need the most, because regulation, especially phobic regulation against genetic technologies, is the biggest barrier to us helping ecosystems. There are amazing innovations out there that could change the world, much like that recombinant factor C for the horseshoe crab blood substitute. And it's been around for 25 years, but it's regulatory and socio-political issues that have made that lag behind. So yeah, there's a way for everyone to get involved, especially if they're creative. As I said at the beginning, Revive and Restore is a nonprofit, so we believe in open source, transparency, inclusion, and as a nonprofit, that's how we steer everything forward. So we rely on donations, but as a nonprofit, we also want to see that citizen movement rise up and that paradigm shift where lots of people are thinking of how they can get involved in their own communities and steer conservation forward. And biotechnology is not a panacea. That's what I should also really say, is protecting habitat, just enacting laws that help species. Those are the real foundations of conservation that have saved species for hundreds of years. And those are the types of things we need continued into the future, where we see biotechnology as how can we use it to improve all of these tried and true conservation methods, and where can we see biotechnology bring new tools to accomplish things that those old methods have never been able to accomplish yet. So we work with everyone from bioethicists to conservation scientists to conservation practitioners to environmental lawyers, to biomedical scientists, people in agriculture, people that have worked in private industries, academic laboratories, even other nonprofits such as Island Conservation, who is an amazing nonprofit that's done great work over the world's islands. We have a huge network of people and scientists that we work with to steer this forward. And if you really delve into it, you'll see that the last 5 to 6 years of dialogue about genetic engineering and gene editing and biotech for conservation has a small Revive and Restore footprint, little watermark on it that's always kind of in the corner of bringing this stuff to the forefront. And I think that's something we're really proud of at the office, and we want to keep going and continuing. We're a small group. There's only 6 of us in the office. We're a startup that allows us to be very flexible. But, for those of you that are emailing us, like, do we have internships or work opportunities? We're too small yet to have those. And that's why I say go to the literature, go to our Ocean Genomics Horizon Scan, go to our website, our publications, and see who our partners are. And those are the people you could possibly reach out to for work opportunities and experience that would get you into this field.
Okay. Awesome. That's great advice for you guys. There you go. So if you you should, and if you're interested in following Ben's Twitter and Revive Restore Twitter, you can do command guest in the chat. It'll link everything. I think it also links the website. And Danny linked it before. Yeah. So command guest, command org, I think, for Revive Restore. So that's their website. Okay. We covered a lot. That was awesome. Thank you so much. Chat, thank you for all of your questions, and thank you for hanging out and listening. Looks like we're finishing today at $1,308.01.
Wow.
Which is awesome.
Here.
Thank you.
The one last thing I will show, to kind of really address some of those questions about ramifications and whatnot. And here, I've got to grab it here. It's actually I had to prop up my computer with a bunch of books. Otherwise, you would have been staring at my nose. So we've published a paper on de-extinction. You can look that up. That's open source. Some of our papers on passenger pigeons are open source. Not every paper we've put out there is open source because of some of the partners we've worked with. It always hasn't been able to play out that way. But we are covering everything from in this field. And so one of the pieces of literature I'm really proud of right now is on animal welfare. It's in this book. the Routledge Handbook of Animal Ethics.
Okay.
And chapter 21 is a chapter I put together. The whole book has a lot of amazing stuff in it. I don't know if you can, not a lot of people are going to afford the price tag on this textbook. It's like $180 or $220. But there's probably ways we can get some of the chapters out to people. Tell your professors about it.
Yeah.
Get it for class. Maybe get it. Sell it back. That kind of thing. But chapter 21 is all about de-extinction and animal welfare. So we're trying to really be comprehensive about thinking about this whole thing, and that's where conservationists are when they breed condors in captivity at San Diego, or when they breed black-footed ferrets at the National Black-footed Ferret Conservation Center in Fort Collins. Everything from animal welfare to the bioethics of what they're doing to the social ramifications of the stakeholders they work with. Conservation is this incredibly transdisciplinary field. And there's something for everybody. We've got our toes in a little bit of it all. And so that's the last book I will tote for our discussion.
All right. Saito, if you could include that in the book list and paste that a couple more times, that would be wonderful. So everybody has those. King with $1.01 and bottle with $1.01. So $1,315.02 then. Okay, everybody's donating, trying to round it out.
All right.
We're gonna round it out, I think.
Someone do 98 cents.
Okay, guys, great questions again. Ben, thank you so much for coming on and talking about this. This is so cool. And
well, thanks for having me. I'm happy to, these are projects that'll go on for years. We have a lot of expanding projects. I mean, I'm happy to come on anytime and tell you more about how biotechnology can change conservation, what cool. Other projects are doing. I mean, there's a lot of other projects out there, some that predate Revive and Restore coming onto the scene that are revolutionizing this field. The world's first actual genetically engineered wild organism is the American chestnut tree. You can look up that story. The State University of New York has a team that's been working on that for almost 30 years. And it's a tree that used to be like the redwoods of the East. In the Appalachian Mountains, it was 1 in every 4 trees. It was this super abundant tree. It's where the song from Christmas, chestnuts roasting on an open fire, it's where it comes from. And once again, just like black-footed ferrets getting hit by plague from Asia, people brought in Asian chestnut trees in the late 1800s. It spread a fungal disease that wiped out the American chestnut tree. And only a few of them managed to survive up in northern areas where the disease didn't reach. And they've been able to splice in a gene from wheat. They found out what causes the fungus to kill the tree. They were able to splice in a gene that actually doesn't even harm fungus. It just prevents the fungus from killing the tree. And they now have American chestnuts growing in greenhouses that have tested to be 100% immune to the fungus. And that's so cool. They're going through regulatory procedures right now, which is a hairy nightmare because no real agency knows how to handle it. But they're going through the regulatory hurdles right now so that people might be able to plant these trees in their backyards, in their national parks and national forests. And in the next few decades, we might have a forest again that has millions of chestnut trees thanks to their work.
That's so cool.
It shows the real promise that these biotechnologies have. And that's old first generation biotech. Now we have gene editing and brand new stuff that could rapidly diversify it. So, I mean, that's work by Bill Powell at SUNY to definitely look into and support as well. He's part of our network of kind of global thinkers on these topics. Yeah, I mean, there's so much going on now that we could have covered. But yeah, happy to come on and talk more about genetic rescue, evolution, and genomics.
Thank you so much. That's so cool. This was different for us. This was really, really cool. Yeah, I'm trying to think. I have so much in my head right now. Like, definitely have more questions, but
well,
most
if
chat's
viewers
questions
have more questions, if some of the viewers have more questions, you have more questions. We're a small office, but we're not impossible to get in touch with. I'm not Neil deGrasse Tyson. I don't have agents handling my media or anything. So you can email questions. Our email addresses are on Revive and Restore's webpage. You can get in touch with us. We might not always be able to get back to your email because sometimes we do get a lot of them and they get buried, but I try as hard as I can to answer most, if not all, the majority of the emails that we get from interested parties and answer some of these questions. So yeah, you can get a hold of us and check us out. Thanks for donating. It touches me to the bottom of my heart. This is working on the passenger pigeon. If you read up on me at all in any of the there's a good chapter in Maureen O'Connor's book called Resurrection Science, all about passenger pigeons and my personal history. I actually have a personal history published in that book, dealing with depression and other mental struggles that many people have dealt with from a young age. And this work has been my dream since I was 12 years old. It's incredible that I actually ended up being lucky enough to get this work as an adult, that the technology came into place. I thought it was going to be still into the future. And so I'm living my childhood passion. And it's changed a lot over the years. How I viewed it as a kid with my naivety is very different from how I became an adult, trying to bring a sense of maturity into it. But I'm living my passion. I hope everyone, gets the chance to really live their passion. And, every dollar you guys put in, it's not only does it bring back passenger pigeons, but it feeds my little boys and it keeps me going. So thank you very much.
That's wonderful. Donations have kept coming in. They're arguing over the cent amount mostly. Abby donated $100. Abby, thank you so much. She said, "Thank you for this stream. It's both interesting and so very important." Thank you, Abby. So that's awesome. Thank you so much. I really appreciate it. Chat, well done today. We learned a lot. Bottle with another $22.75. Thank you if you donated today, if you subbed today. I appreciate it. Connor with $30. So we hit our 1,500. We're at $1,531.75. So guys, awesome. Thank you so much. Ben, thank you so much for your time. I'll be in touch. And guys, go follow Ben's Twitter and Revive and Restore's Twitter, and I'll keep you posted if I see stuff and you don't. And maybe down the line, if we have if you guys have your breakthroughs in the next couple years, then we'll have lots to talk about.
Yeah.
That's super exciting.
Thanks for introducing me to all your viewers. It's an incredible community. And I'm not as savvy on all this stuff. My gray hair is coming in. I don't do Twitch or anything like that, so I don't even have time anymore. So this is awesome. This is an awesome chance to get to reach a new audience. Thank you very much, and thanks everyone.
Oh, of course. Thank you. Okay, I'll speak with you soon then.
All right.
All
bye.
Right, bye.